Okay, for the continuation of our life. So what is the need of cell division? In order to continue our life, we need cell division. So does the cell division happens every time? No. The cell division happens only when there is need. For example, you think that if cell division happens continuously, okay, if the cell division happens continuously, we are going. Okay, if cell division happens continuously, we are growing day by day. Every day we will grow. Okay, we can grow up to the sky. But here, the cell division is limited. When there is need only, the cell will divide. The cell will divide. So, cell division happens in all the living organisms, whether it is plants, animals, or microorganisms. Microorganisms, the cell division happens. So, we are discussing about mitosis. Okay, so generally mitosis is the division of one cell into two cells. Division of one cell into two cells. 1 to 2. And the first cell we call it as parent cell. Parent cell, and these we call as daughter cells. Daughter cells. In the previous topic itself, we have discussed about interphase. 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 Okay. Phase means stage. Inter means in between. So from one mitosis to another mitosis, the gap which is happening with one mitosis and to the next mitosis. For example, one cell is present. It is divided into two. Okay. And these two cells, this again two cells. Again they will give to two. Okay, so two cells are formed. Again this cell should give two cells. So this gap it is known as interphase. So one mitosis to another mitosis. The time period between one mitosis to another mitosis is known as interphase. Mitosis is nothing but only the cells will divide. Only the cells will divide. Nothing will divide. The cells will be, the one will be going into two. No division happens. One is moving to two, but no replication, doubling happens. But the time which is from one mitosis to another mitosis, what is happening is the interphase. We already have discussed. In the interphase, we have what? What are the phases we have? G1 phase, S phase, G2 phase. G1 phase, S phase, G2 phase. For example, one cell is given 2. So this should also give again 2. So what is happening in that cell? In that cell, what is happening? Cell size increases. What is happening at G1 phase? Cell size increases and DNA starts replicating. DNA starts replicating. So starting, what is the chromosomes are doing? The chromosomes are producing exact copy of that chromosome. Start at this G1 stage. Okay, cell size becomes big. And what the DNA is doing? It is producing its replica. Replica means what? Carbon copy, same as it is. They are starting to form. And in S phase nothing but synthesis. Synthesis means what? For example, here DNA 2 are present, 2 chromosomes are present, for example. 2 are started to produce again 2. Here what is happening? 2 produced 2. 2 has produced 2. So here G2 phase, it stops the replication. Stops the replication. Means what? Here starting of DNA replication, starting. Synthesis means total are produced. How many are needed? That are produced. If 2 are present, 4 are produced here. For example, in human beings, 23 pairs are present now. Again, another 23 pairs are synthesized here. Here started, here over, and here stop. If it does not stop here, what happens? The 23 pairs are produced again 23 and they will start to produce again and again. Okay, it is disturbance of chromosomes. So here starting, here total produce, here stop. G1, S, G2. So already we have discussed in the previous topic, but it is now it is important. What is happening here? Interphase means from one cell to another cell mitosis. In between the phase, the DNA replication happens. That phase is known as interphase. Again, the interphase is divided into three stages. Okay, three stages: G1 phase, S phase, G2. G1 means cell size increase, DNA replication starts. Yes, synthesis. Synthesis completed. Total chromosomes completed. G2, stopping of the formation of chromosomes. This is the thing happening. So now we can say at the end of the G2, what is the condition of that cell? At the starting of the G1, the condition of the cell is small with only one set of chromosomes. Okay, once again I am repeating. So one cell has divided into two. So this cell, I am taking the example of this cell. So at the starting of interphase G1, what is the condition of the cell? Small, and only one set of chromosomes are present. Now at the end of the G2, what is the condition of the cell? It is increased in size, means double the size we can say. Double size and double number of chromosomes. Double set of chromosomes. Understand? So what is present starting? Only one set. Size of the cell is small. At the end of the interphase, size is big, double the size and double the number of chromosomes. So what here we should do? One cell should be divided into two cells. So what here double it has present, no? It should share to two daughter cells. How it is sharing? Directly it won't go, no? The cell won't say, you one side you go that side, one side you come this side. It won't say, because in this double set, one is original copy, one is duplicate copy. Duplicate, replica, nothing but same as it is. You can't detect whether it is original or duplicate. Okay, but here at the end of the G2 phase, what is the condition of chromosomes? One is original copy and one another one is which is produced from the original copy. Okay, so how the sharing is happening? Let us discuss in this. What is this mitosis? Mitosis. Please write it in your notes. Okay, so listen here, students. So before going to the mitosis division, let us discuss what are present inside a cell. For example, if it is a cell, it is what cell membrane. Cell membrane. Inside what is present? Nucleus is present. Inside the nucleus, okay. Inside what is present? Nucleolus is present. Around this, genetic material is present. These are nothing but chromosomes. Okay. Once again, I am repeating. This is a cell because why I am highlighting nucleus means the nucleus only responsible for the cell division. Okay, inside the nucleus, what are present? Genetic material is present. How the genetic material is? It is thread-like structure, colored bodies, thread-like. How the threads? They are colorful threads. They are spread even like this. And another one body is present. This is known as nucleolus. What is this? Nucleolus. This is what cell membrane and nucleus. Inside what are present? Chromosomes are present and nucleolus is present. And now that the starting of the mitosis, this cell size is very big and chromosomes are double. Chromosomes are double. This is the condition. First condition means what? First stage to start with the mitosis. So now the cell is ready to go to participate in the mitosis. Means the cell is ready to divide into two. Okay, it is participating in the mitosis. Cell size double and chromosomes are double. Okay, now it is ready to divide. Means it is ready to go with the mitosis. So mitosis again it has 4 phases. It is again have 4 phases. First one is prophase. Okay, what is the first stage? Prophase. In prophase, okay, in prophase, what is happening means these chromosomes, okay, chromosomes are getting contract. Chromosomes are getting what? Contract. Because the chromosomes in the nucleus, the chromosomes are spread like this, no? So we can't take it as means it is in a zigzag manner. But now in this phase, what is happening? The chromosomes are getting contract. Okay, chromosomes are getting contracts and they are becoming spiral. Spiral nothing but like this. Okay, they are becoming what? Spiral. And next, inside this, what is present? Nucleolus is present. No, it is becoming very small. The nucleolus will become small. The nucleolus will become small. Next, around the this is what? Nucleus. Okay, this is what? Nucleus. This is what we are nuclear membrane. Around the nucleus, what is present? Nuclear membrane is present. So at this stage, what is happening? The nuclear membrane slowly disappears. Okay, nuclear membrane slowly disappears. Okay, this is the cell. If you consider it as a cell, and this is the nucleus, and this is the nucleolus. Okay, so what is happening at this stage? Nuclear membrane disappears. What is happening? Nuclear membrane disappears. Now they will form. What is that? Centromeres like this. You can see, I think so. This. They will be like this, no? They will form V shape like this. Okay, chromosomes. Okay, this only we can see in the animal cells. Animal cells. So what is happening here? Chromosomes, they are forming into spiral shape, and the nucleolus is becoming very small, and nuclear membrane disappears. Nuclear membrane disappears. Now it is forming what? With centrosome. With the centrosome, and the chromosomes are forming into chromatids. These are known as chromatids. Before chromatids, how they are thread-like structures. Now at the end of prophase, what is the structure? The chromosomes are changed into chromatids. Changed into chromatids. So this is the prophase. So please write what is that? Chromosomes are forming into spiral structures. Nucleolus will become small. Nuclear membrane disappears. Chromosomes are arranged like this in the form of chromatids. So next, so after this, what is that? Metaphase. Second one is metaphase. At metaphase, spindle fibers are formed. Spindle fibers are formed. Second stage, what are formed? Spindle fibers are formed. So spindle fibers nothing but they are formed like in the equator. So for example, these are the chromosomes with centromeres. So what are formed? So they are attached to the spindle fibers. So this side, one side, this side. So if the cell is okay, so see here. So this is known as what? Equator, no? We will call it as what? Equator. Equator nothing but which is dividing midline of the circle is known as equator. So centromeres are at the equator with the spindle fibers. So in the metaphase, what are produced? Spindle fibers are produced. Spindle fibers are produced. And next, third one is anaphase. So in the anaphase, what is happening? These spindle fibers move one chromatid to this side and another chromatid to this side. Understand? So each chromosome is having two chromatids, no? So in the second metaphase, what they have formed? They have formed spindle fibers. And in the third stage, anaphase, what they do? With the help of these spindle fibers, they move one chromatid to this side and one chromatid to this towards the poles. If it is equator, this happens what? Poles. So one spindle fibers to spindle fiber to another pole. So this is happening. What is that? Two poles. In anaphase, what is happening? They are moving the chromatids to one pole and towards the another pole. So what is happening here? So this side one set of chromatids. This side one set of chromatids. Means what? Now the double chromosomes are moved single towards one pole and single towards another pole. So we will call as haploid. What we call as haploid. Diploid means double. Diploid means double. Here the diploid. Now at the anaphase, what is happened? Haploid. Means half towards one pole and half towards another pole. And next one is telophase. Last phase is what? Telophase. Telophase. So what is happened? What has happened in the anaphase stage? One set towards one pole and another set towards another pole. So at this time, towards one set around the chromatids, nuclear membrane comes, reappears. Nuclear membrane reappears. For example, this is a cell. Okay, nuclear membrane already disappeared in the prophase. In the second phase, third phase, there is no nuclear membrane. Now what happened here? So towards the poles, what was present? Chromatids are present. Now nuclear membrane reappears where the chromatids are present, like this. Okay, so how many nuclear membranes formed here? At one pole, at another pole. So two. How many formed now? Two. In the prophase, one nuclear membrane disappeared. But here nuclear membranes, how many? Two nuclear membranes. And after that, this cell membrane will pinch, contract like this. And slowly it comes towards the equator. And these two will be divided into two cells. So at last, what was happened? Two nucleus with two cells. Okay, two nucleus with two cells. And at the end of the mitosis, we will see what? Two daughter cells with equal number of chromosomes present in the parent cell. And again, all mitochondria, endoplasmic reticulum, they all again reappear when nuclear membrane reappears. Okay, so what happened? First, nucleus is divided. Nucleus has been divided. Nuclear membrane divided. And next, what was divided? Cell membrane divided. So first, what was dividing? Karyokinesis. Karyokinesis nothing but nuclear membrane is divided. Second, what is dividing? What is that? Cell membrane is dividing. That is known as cytokinesis. So you will be asked the question. First, in the cell division, which division happens in the first? What is that? Division of the nucleus. Secondly, what is that? Division of the cell membrane. Division of the cell membrane. This is about telophase. So, what is that? Prophase, metaphase, anaphase, and telophase. So, in prophase, what is happening? Nuclear membrane disappears. Nucleolus becomes small size. Chromosomes will become chromatids. In the metaphase, what is happening? Spindle fibers comes. In the third anaphase, what is happening? That spindle fibers are moving chromatids towards the poles. In the fourth telophase, what is happening? Towards the poles, nuclear membrane is appearing. Nuclear membrane is appearing. So what was happened? Two nucleus formed. After the total formation of two nucleus, the cell membrane pinches or punches towards the middle, dividing the two cells. So one cell has been divided into two cells with equal number of chromosomes. So please make it a